The Hershey-Chase experiment. Alfred Hershey and Martha Chase helped to confirm that DNA was genetic material in 1952. How did they do this? They did this by conducting two side-by-side experiments with separate bacteriophage, or viruses, cultures in which either the protein capsule is labeled with radioactive sulfur or the DNA core is labeled with radioactive phosphorus. There were 3 steps to this: infection, blending, and centrifugation. First, the radioactively labeled phages are then allowed to infect bacteria. Second, agitation in a blender dislodges phage particles from bacterial cells. Lastly, the centrifugation concentrates cells, separating them from the phage particles left in the supernatant. The results of this were: first, radioactive sulfur is found predominantly in the supernatant. Second, radioactive phosphorus is found predominantly in the cell fraction, from which a new generation of infective phage can be isolated. The conclusion of this experiment was the active component of the bacteriophage that transmits the infective characteristics is the DNA, showing a clear correlation between DNA and genetic information. Many scientists of the time thought that proteins carried the genetic information, but this was disproven by the Hershey-Chase experiment.